Hello students, today we will study about biosynthesis of purine nucleotides. There are two types of nucleotide, that is purine and pyrimidine. Purine type of nucleotides available in our body are adenine and guanine, whereas pyrimidine exists in the form of cytosine, uracil and thymine. The difference between purine and pyrimidine is the purine is a double ring structure and also it is numbered in the anti-clockwise direction. You can see here this nitrogen is given the first number, second, third, fourth, 5, 6, 7, 8, 9. Similarly, in pyrimidine the numbering is in the clockwise direction, that is first number to this nitrogen, second, third, fourth, 5 and 6. So this is the difference between purine and pyrimidine. Now you are aware about the structure of purine. Purine is a double ring structure with a imidazole ring in it. Now we will go through the biosynthesis of this purine nucleotide. This is the purine nucleotide biosynthesis. Now the precursor for the synthesis here is a ribose 5-phosphate. You can see here the alpha D ribose 5-phosphate, which is the pentose sugar containing the phosphate group at the 5th carbon, is the precursor for the synthesis of purine. You can see here phosphate group at the 5th carbon, therefore ribose 5-phosphate. Now this ribose 5-phosphate get converted to 5-phosphoribosyl alpha pyrophosphate. Means this pyrophosphate group is attached on this hydroxyl group with the help of enzyme pyrophosphate synthetase, and it is accompanied by the conversion of ATP, means ATP is required for this conversion. Now this 5-phosphoribosyl alpha pyrophosphate get converted to 5-phosphoribosylamine. Here you can see ribosylamine, it is getting converted to ribosylamine, means this pyrophosphate group is detached or removed from here and the amino group is attached here. And this amino groups comes from glutamine. So wherever for the synthesis of this purine nucleotide amino group is required, it will come from glutamine amino acid. Right? Now the enzyme required here is phosphoribosyl pyrophosphate glutamyl amidotransferase. Glutamyl amidotransferase. Now the product form here is 5-phosphoribosylamine. In the next step, this phosphoribosylamine gets converted to glycinamide ribosyl 5-phosphate. Now glycinamide, as the name suggests, the glycine enters here and with the help of synthetase enzyme and ATP, this glycine gets attached to the amino group to form glycinamide ribosyl 5-phosphate. Now in the next step, formyl group is attached, this is the formyl group which is attached to the amino group of the glycine which is previously attached to this glycinamide ribosyl 5-phosphate, and this formyl group comes from N10-formyl tetrahydrofolate which gets converted to tetrahydrofolate and donates its formyl group to this glycine. So the product form here is formyl glycinamide ribosyl 5-phosphate. Now formyl glycinamide ribosyl 5-phosphate in the next step gets converted to formyl glycinamidine ribosyl 5-phosphate. This imidine means attachment of again amino group at this carbon. So this here the entry of amino group is again, therefore it comes from where? Glutamine. Glutamine is a amino acid which provides this amino group here. And the enzyme required for this step is synthetase and ATP is utilized. This formylglycinamidine ribosyl 5-phosphate gets converted to 5-aminoimidazole ribosyl 5-phosphate. Means here the cyclization means imidazole ring is formed. Here the ATP is required. ATP gets converted to ADP and this cyclization takes place. 5-aminoimidazole ribosyl 5-phosphate then undergo carboxylation. Carboxylation means entry of carbon dioxide. This carbon dioxide with the help of enzyme carboxylase get attached to the fourth carbon of this imidazole ring to form 5-aminoimidazole carboxylate ribosyl 5-phosphate. This imidazole gets converted to 5-aminoimidazole carboxylate. After conversion of this, the aspartate group enters, that is aspartate which is the amino acid which enters and it gets attached to this carboxylate group. This carboxy group aspartate get attached here to produce 5-aminoimidazole 4-succinyl carboxamide. This is 4-succinyl carboxamide group. In the next step, this 5-aminoimidazole 4-succinyl carboxamide ribosyl 5-phosphate removes the fumarate from that and with the help of enzyme adenosuccinate lyase and produce 5-aminoimidazole 4-carboxamide ribosyl 5-phosphate. Here you can see only CONH2 is remaining and rest of the chain, carbon chain is removed in the form of fumarate. Again in the next step, formyl group is attached which comes from N10 formyl tetrahydrofolate which gets converted to tetrahydrofolate and with the help of enzyme formyl transferase it gets converted to 5-formamino. Means this amino group is getting attached to this amino group. So 5-formaminoimidazole 4-carboxamide ribosyl 5-phosphate is formed. Now this is ready for cyclization. So with the help of enzyme cyclohydrolase and dehydration from this means H2O is removed to produce here inosine monophosphate and this is our basic purine nucleotide. This is the basic structure for nucleotide. You may feel this cycle difficult to remember. So for that purpose you must know what is actually happening during the synthesis of inosine monophosphate. You can see here in the first step there is only the activation. Means with the help of pyrophosphate here this site is activated and after activation of this site the glutamine enters and provides its amino group to attach at this site. So first step is this activation and the next step we need the amino group, therefore glutamine is entered in the first step. In the second step glycine is entered which attached to this amino group to produce glycinamide. Then in the third step we require the formyl group which gets attached to the amino group of glycine to produce formyl glycinamide. After the entry of formyl group in the third step, in the fourth step again the glutamine is entered to provide this amino group which attach to the carbon and then in the next step the cyclization takes place. So this is the first cyclization after entry of glutamine, glycine, formyl and again glutamine. The first cycle or first ring is completed. After that to produce the second ring again the carboxylation takes place. This carboxylation takes place with the help of CO2. Here at the fourth carbon, this CO2 gets attached, and after that aspartate is entered to attach this carboxyl group and provide the amino group for this next ring. After that aspartate gets removed in the form of fumarate, leaving its amino group there only. And after this amino group is attached here, now again the formyl group is required to increase the number of carbons. So with the help of formyl transferase, again this formyl group is attached to the amino group present here, and in the last step with the help of cyclohydrolase, this ring is completed and cyclization here takes place to produce inosyl monophosphate. So just remember the steps. First step is activation, second is entry of glutamine, third is glycine, fourth is formyl, fifth is again glutamine, and sixth is cyclization of the first ring. Then the carboxylation to increase the number of carbon, then the entry of aspartate. After entry of aspartate, there is removal of fumarate, leaving behind the amino group here, then entry of formyl, then again the cyclization or secondary formation, and finally we get the inosyl monophosphate, which is the basic nucleotide molecule, purine nucleotide molecule. Hope now that this cycle is clear to you all. Thank you.